طيب السلام عليكم. أنا I think all right. Yeah, thank you. First of all, I'd like to introduce myself. So my name is Amira Mitwali, and I am a lecturer in microbiology and immunology at Technical University Munich. In the beginning, I'd like to thank Dr. Hadidi for inviting me to give this talk today, and I would like to thank all of you for being here. So as you can see, the title of my talk is Gut Microbiome Signatures in Health and Disease and Moving Beyond Correlations. All right, so just to give you a little bit an overview of what we will discuss today. So first of all, I will give you a very brief introduction about human gut microbiome. Next, we move to gut microbiome biomarker discovery, which would be the main topic of today's lecture. And finally, the focus of this lecture will be on the implications of microbiome signatures. And for that, I will show you one example of a showcase. So we have what I have been working on most of my career for the last 7 to 8 years was inflammatory bowel disease and the role of gut bacteria in inflammatory bowel diseases. I think how we would organize this lecture would be, it would be ideal if you can keep all your questions to the end of the lecture. So you can either write it down in the chat or you can just keep it noted down until the end of the lecture. I hope I will try to keep at least 10 to 15 minutes for discussion in the end to address any of your questions. All right. As far as I know, this lecture is the last one in a series of lectures on gut microbiome. So I think if you have been around for the very few lectures, I think you're well introduced to gut microbiome. So I'm not gonna go in length about that, oops. Yeah, all right. Okay, so as you probably know now, microbiome or microbiota is a term which comprises all the microorganisms in our body. So that comprises bacteria, fungi, viruses, and all the microorganisms. And if you, just to give you a little bit of statistics about the gut microbiota or the microbiota in general in our bodies. We have in our bodies around 10 trillion human cells. However, we have around 100 trillion microbial cells, which means we have almost 10-fold more microbes than our human cells. So we are mostly microbes, or we are actually mostly microbes compared to human. And if you think that, okay, so it's actually the DNA which matters, if you look at the number of genes, you will see that we have around 20,000 human genes in our bodies. And in contrast, we have around 2 to 20 million microbial genes. So again, we are outnumbered by microbes in any case, in terms of number of cells or number of genes. So all these microbes encode a lot of genes, which in turn encode functions that are important to our bodies, to our physiology and homeostasis. And so that's just to give you a little bit of impression how important our microbes are. And the second term, which we will also use quite frequently during the lecture today, would be microbiome signatures or biomarkers. And when we speak about microbiome signatures, what we actually mean is a unique pattern of microbiome configuration. So that means that we're not describing one or two bacteria, but we actually describe the whole community. And in terms of relative abundance, in terms of presence and absence, in terms of functional capacity and functional potential. And these signatures or biomarkers should be able to stratify defined physiological or pathological conditions, and they should be used to predict the risk of disease development or progression, or should be capable of doing that at least. And as you know, an ideal biomarker should be quick, should be non-invasive, should be objective, and should be accurate in predicting a certain disease outcome, for example. And so that's what we mean by microbiome signature. So the aim is, or what we have, what I mainly focused on during my research is to define microbiome signatures that can be used to identify certain pathological conditions, and that can be used to predict the risk of relapse or the risk of disease development in mainly in inflammatory bowel diseases. All right. So as I told you, I introduced you to the human microbiome in general, but actually the focus of today would be the human gut microbiome. And the human gut microbiome makes around 70% of all the bacterial population in our body. And if you look at the human digestive tract, you will see that our digestive tract harbors a complex array of microorganisms. And this goes here, as you can see, in a gradient of complexity. So there is an increase in the number of bacteria along the GIT from the stomach down to the ileum, and it reaches the highest density in the colon. And overall, we have two dominant phyla, so the Firmicutes and Bacteroidetes in our gut microbes. And they also comprise around 1,000 unique species. And so along the GIT, we also have a gradient of pH. So the pH gradient goes from low pH up to higher pH along the colon. And that actually allows for the enrichment and for the localization of certain bacteria in each location, and in turn would affect which bacteria would be able to colonize which compartment along the GIT. All right. All right, so have you ever thought how did we acquire our gut microbes? So actually we acquire our gut microbes from the moment we're born, and until we die, we have our microbes. And the way or the mode of delivery, how we're born, would impact which microbes would colonize our bodies, and which bacteria would shape our gut microbiome. And so, for example, babies who are born naturally or via vaginal delivery, they would have the gut microbes similar to the microbes which are in the birth canal of the mothers, while babies who are born via section would mostly harbor microbes from similar to the skin microbiota in the mother. So that in that way, we already have different microbiota when we are born. And along our life, different factors can impact and can shape our gut microbes. And these factors can include, for example, the use of medications. So in particular, antibiotics would impact the community composition and community diversity of our microbes. Also, diet, of course, would be one very important factor which impact the gut microbiome composition and function, host genetics as well, while there's no real understanding of how host genetic would be causal for changes in gut microbiome. But there are already links of host genetics to changes in gut microbiome. We can touch on that later, but data from twin studies, for example, show or people who are raised in the same, people from the same families, related individuals would have similar microbes compared to unrelated individuals, for example. And of course, environment is a very important factor. So there are a lot of studies which show that people who are living in industrialized countries compared to rural environments, for example, would have a very different gut microbiome composition. And so during the first 3 years of life is what we call a window of opportunity. So during this time, all these factors can impact or can shape the gut microbiome composition. And once you reach an age of 3 years of age, you will reach a gut microbiome configuration which is similar to an adult microbiota, and that would be the microbiota that would stay with you for life. So unless you have a very dramatic change in environment, in your dietary habits, or in the medications you take, your gut microbiota should be stable during your life. And during these 2-3 years of your life, all these factors can impact the gut microbiome composition. All right. So there have been a lot of, so people have been studying microbes for ages. And in the beginning, people used, of course, classical or conventional or very primitive methods like microscopy or plating bacteria on agar and isolation and identification of bacteria. However, there have been in the early 90s, there were actually the late 90s, there was the genomic revolution. So there have been a lot of studies using next generation sequencing, so shotgun metagenomics or 16S RNA gene sequencing and other methods to study human microbiome. And one of these efforts was, of course, the Human Microbiome Project, which was funded by the NIH in the United States. And this project was, or actually in this project, they sequenced the microbiome from different locations in human bodies from so many individuals. And they looked at the gut microbiome, or not only the gut, but the human microbiome in general in different locations. And they looked also at the functions of microbes. And so one of the main findings of the Human Microbiome projects, there have been a number of milestone findings, but one of the main findings here was looking at the diversity and the stability of microbiome within a healthy population. Of course, we were all keen on studying the microbiome composition in disease, but to be able to understand the relevance of microbiome in disease, we need first to define how a healthy microbiome looks like. And so we would be able to quantify the changes which happen in disease, right? So one of the changes or one of the conclusions of the Human Microbiome Project was that we are not all equal, right? So if you look at this figure here, it might look complicated, but actually it's not. So let's look at here and consider each line here is one individual, and each square would describe either the diversity or the function of microbiome in one ecosystem. So oral microbiome, stool, in different compartments, in different body sites. And what you can see here, so that was the microbiome composition in terms of taxonomic composition, taxonomic diversity. And what you can see here is that we humans are diverse in terms of taxonomic composition of our microbiome. So, which means you can have some individuals who have, for example, here, a high firmicutes composition compared to proteobacteria in yellow, while you have some people who would have the other way around, so they would have a dominance of proteobacteria compared to firmicutes, for example. And this can be also seen in other compartments. So we have a huge diversity in our gut or in our, I'm sorry, I'm always stuck to the gut because that's always what I work on, but actually here it's all body sites, so different body sites. All right. So then on the other hand, when we look at the function, so here they did shotgun metagenomics of all these body site samples from different humans in this project. And what you can see is the opposite picture. So you can see that in all humans, we have very similar functions. So you can see that we're almost identical, right? So you have a slight variation, but in general, we are very similar in terms of the functions which our microbes can perform. And that's a very important feature of the microbiome, human microbiome, which is termed as functional redundancy. So, of course, we all need eventually the same functions to maintain the homeostasis in the body, to carry out the same physiological functions. So it doesn't matter if you have different bacteria which reside in your body. What's important is that these bacteria can eventually perform the same functions. Okay. So just to make sure that you are following, are there any questions up to now? Okay. Okay. So another thing which is very important when we talk about microbiome is that the lifestyle and environment can impact the microbiome composition. As I just told you earlier, that there have been a lot of studies which looked at different populations in, for example, in industrial countries or in rural countries, and comparing the microbiome composition, and there was drastic differences between both. And these changes are, of course, driven by many factors, so including genetics, including the diet, which is the dietary habits in each country. For example, in industrialized countries compared to rural areas. And here, also, the geography or the location has been identified as one of the major variables which contribute to changes in microbiome composition. And in this recent review that we have published, we looked at CNS RNA gene sequencing data in multiple populations across the globe. And the aim was to see if we have, or if all the published data sets can come up with a unique signature which can identify or which can be used as a biomarker to identify inflammatory bowel disease or type 2 diabetes. And as you can see here in this picture, you can see the map and you can see the different locations we have looked at, and we have looked at their data, that we actually don't have really a unique microbiome signature in all these locations. So it's quite variable. And here we identified, for example, the bacterial taxa which would be enriched or decreased in each location, and in inflammatory bowel disease or in type 2 diabetes. And what we can see is that we have a very heterogeneous picture. So we have a little overlap. So some taxa were always coming up in different locations, but in general, there was a huge variability between different locations across the globe. And so to identify a unique microbiome signature, which can be used in different, across different populations, across different ethnicities, it's not really an easy task. And apparently it's not enough to look at the gut microbiome or the microbiome diversity, taxonomic diversity. What we need to look at is also the function. So we need to move beyond the simple correlations or associations, and we need to understand the functions of these bacteria, and then we can understand the causal link to different diseases. And so if you look at how, I mean, with this genomic revolution and with the ability to sequence a lot of samples at the same time in a high throughput fashion and to do all these multi-omic analyses. So I think you've been introduced to these, to metabolomics, shotgun metagenomics, and all these tools in the previous lectures. So the ability to use all these techniques have yielded a lot of data around microbiome and the relevance to different diseases. And so in this figure here, what you can see is the number of publications per year, and stratified by the different disease entities. And what you can see is that over the year, there have been more and more data and more publications generated around the link between microbiome and different diseases, in particular chronic diseases like IBD, inflammatory bowel disease, but cancer and obesity, infection, diabetes, allergy, cardiovascular diseases, and brain disorders, of course. So that gives you an impression of how growing this field of microbiome is, and also on the relevance of microbiome to different diseases. All right, so as I told you briefly, there are different ways to study microbiome, and I'm sure you've been introduced to that in the previous lecture. So if you want to look at the composition, you would use 16S RNA gene sequencing. If you want to look at the function, you would need to do the shotgun metagenomics. to look at also strain level diversity and to look at the functions which are encoded in the genes that are encoded by these bacteria. So I'm not going to go in details here. What I really want to introduce you to is another tool that we use to study the gut microbiome and their causal link to disease. And this is what we call gnotobiology, or the use of germ-free mice. And so just to introduce you to that, to this tool, here we use germ-free mice. So these are mice which are raised in a completely sterile environment. So they are completely devoid of bacteria. They don't have any bacteria in the gut and the mouth and on the skin. They are just completely sterile. And these mice are born and raised in a sterile environment. And the beauty of this model is that you can manipulate the mice by inoculating or cultivating certain bacteria which you are interested in. And these bacteria can be of different complexity. So you can either colonize the mice with a single bacteria, that would be a monocolonization. You can have a set of bacteria, which we would call a defined bacterial consortium, or you can have a complex microbial community. So you can take the whole human microbiota and use it to colonize a germ-free mouse. Okay. And this model is really sophisticated. So in these, I can show you now a picture of how a germ-free facility looks like. And this is the germ-free facility that we have at the Technical University in Munich. And this is one of the very few germ-free facilities across Europe. So it's really sophisticated, and it really needs experienced personnel to handle it, because you need to make sure that all the environment is kept sterile, and because one single contamination can ruin your experiment. All right, so here what you can see in these pictures are what we call isolators. And these are basically like plastic incubators. And inside you have the cages where the mice are housed. And inside it's completely sterile. So when I go to this facility, I need to change twice. I need to have a special mask, and I need to work completely in a very sterile, I need to keep everything sterile and to maintain everything sterile. What you do is that you go inside these gloves, and you can handle the mice while they are inside. And of course, you have these isolators are ventilated by, and the air is completely sterile as well. Why I'm introducing this, because I will show you in the paper that we will discuss today that we use this tool to study the functional role of microbiome in inflammatory bowel disease. And as I told you, the beauty of this model is that you can really, if you're interested in one bacteria, you can use it to inoculate the mouse model of interest, and then you can see what happens to the mouse, and you can study the host, microbe-host interactions in the mouse. Okay. Okay. And so just to show you an example of how this mouse model was used, or the germ-free mice are used, the very first study which established a humanized mouse model was done by Geoffrey Gordon in Washington. And what he did, he actually he observed that he has a wild-type mouse and a mouse which has a genetic defect, and this genetic defect leads that the mouse is always would always eat until it becomes morbidly obese. And when he looked at the gut microbiota composition of these mice, he observed that the obese mice would have a gut microbiota composition that is different from the lean mice. And so what he did next to see if the gut microbiota would be responsible for this phenotype is that he isolated, or he extracted the gut microbiota from these two mice, and then he transferred the microbiome into germ-free mice, right? And it was great what he observed, that the germ-free mice which received gut microbiota from a lean mouse were also lean, and the germ-free mice which received obese microbiota became obese as well. And they had, if you look at this PCA, they had also a complete different gut microbiome composition, and they had a difference in the increase in body fat. So this study was proof of concept that germ-free mice can be used to study to look at the causal link of gut microbiome to disease, and to understand or to study microbe-host interactions in a model to validate these changes in microbiome composition in a model in vivo, and to be able to validate it as well. Are there any questions up to this point? All right. Okay, good. So now we've seen that microbiome can be involved in many chronic diseases. And what we need to do now, so we have, I mean, all these efforts in trying to identify and to characterize microbiome using all these technologies, high throughput technologies in sequencing, there have been a lot of data generation. So there are a lot of data available, and all of these data are merely associations or correlations. However, we still need to understand if these changes in gut microbiota in disease, are they causal? Is it a cause or a consequence of disease? Does it happen before the disease takes place, or it's actually a consequence of the disease incidence? And so here we move to the main focus of today's talk. So I'm going to present you the findings of a paper that we published last year in Nature Communications. And in this paper, let's go on. Yes, so now I wanted to present you findings of a study that we published last year in Nature Communications. And in this study, the aim was to identify a microbiome signature which would be unique to inflammatory bowel disease, and that microbiome signature would be used would be able to be used to classify patients based on disease outcome, all right, or based on treatment outcome. And here in this paper, what we did was, first of all, just give you a little bit of an idea about IBD, or inflammatory bowel disease. I think maybe some of you know it already, but if not, just briefly, inflammatory bowel disease is a chronic relapsing condition that affects the gastrointestinal tract, and in inflammatory bowel disease can be classified in two entities. So it's either Crohn's disease or ulcerative colitis. And as you can see, it's pretty nasty. And here you can see how the colon or the gastrointestinal tract looks like in a healthy individual, and how it looks like in Crohn's disease or in ulcerative colitis. So here you can see that there are lots of ulcers, bleeding, and it's really painful, and it affects the quality of life of patients dramatically. So in Crohn's disease, it mainly affects, it can affect any part of the intestinal tract, so from the ileum to the colon. It can have an ileal phenotype, colonic phenotype, or it have an ileocolonic phenotype, so a mix of both. Or the ulcerative colitis is mainly focused to the, let's go on. So we stopped here. I wanted to introduce you to inflammatory bowel disease, and that inflammatory bowel diseases comprises two main entities, Crohn's disease and ulcerative colitis. And in Crohn's disease, it mainly affects, or it can affect any part of the intestinal tract, so going from the small intestine to the colon or the rectum, while ulcerative colitis is usually reserved to the colon. And as you can see here in this colonoscopy, how a healthy colon looks like and how it is impacted in inflammatory bowel diseases. And then there have been a lot of studies around the causal or the causes to inflammatory bowel diseases. And now what's believed is that inflammatory bowel diseases are caused by an interaction between changes in gut microbiota composition and in a genetically susceptible host, and also a compromised immune system. And so in this study, what we wanted to do is to identify an IBD relevant gut microbiome signature using techniques using genomics, so 16 sRNA gene sequencing, shotgun metagenomics, metabolomics, and in addition, we do the functional validation in humanized mice. And so we had this cohort of patients, which was recruited in Spain and a clinic in Barcelona. And this is a very unique cohort of patients. So, you know, inflammatory bowel disease patients are usually treated by biological therapy, so monoclonal antibodies, or also immunosuppressives, corticosteroids, and these are the conventional treatments. However, in some cases, when the patients do not respond to any of these treatments, they can finally be advised to receive hematopoietic stem cell transplantation. And the rationale behind using hematopoietic stem cell transplantation is to do an immune reset so that the immune system would not recognize the gut microbiota as foreign. They would not overreact to the gut microbiota in the intestines, and so there will not be exaggerated immune response. And so this cohort of patients, we had 35 patients, and what we did was that we collected fecal samples from these patients over time. So at baseline before the treatment, and then every 6 months for 5 years after the transplant. And then it was an observational study. So we followed the patients over time, and we formed then groups of patients. So some patients responded to the treatment and they stayed for long-term remission, and some of them actually relapsed after some point, while some other patients did not respond to the treatment at all. And so the idea was to use these tools to study the gut microbiota composition, structure, and function, and to see if we can identify any signatures that can help us predict patients who would be susceptible to disease or patients who had a higher risk of relapse. And so here, that's an example of one patient. And here what we did was, again, collecting fecal samples over time and looking at the gut microbiota composition in these samples. And if you look at the fluctuations or the dynamics in this patient scenario, you will see that this is the patient, this is a sample which was collected from the patient at baseline. So when the patient was in active disease state. And then we followed the patient after he received the treatment over time. And when the patient went in remission, we saw a change in gut microbiota composition. So here, this is a beta diversity analysis. And I think you've been introduced to this analysis before, but simply what you see here, each dot is one sample, and each dot would represent the gut microbiota composition of one sample at one time point. And the distances between these dots would describe the microbial shift. So the further the two dots are, that means that the gut microbiota composition is very different. And when these two are very close, that means that they are very similar or almost identical. Okay, and here what we wanted to look at is how the gut microbiota composition was changing over time and in response to treatment. And so what you can see is here, when the patient went in remission, there was a clear shift in gut microbiota composition. So you can see that it moved here to this point, and the patient stayed in remission. So there were some changes here, but they were not so dramatic. And then here at time 0.7, the patient relapsed. And you can see, I hope you can appreciate how the change is here between time 0.7 and 8. So there was a dramatic shift in gut microbiota composition when the patient went in relapse. And so in essence, that means that there are fluctuations along the disease course, and that the fluctuations or the changes in gut microbiota composition correlate with disease severity. Okay, and then we wanted to look at the full cohort of patients, and here again, can we? Second. All right. So what we wanted to do here is to look at the full cohort again and to see how the microbiome composition or microbiome profiles look like. And in red here, we have all patients or all samples which are from patients that are in active disease. And in blue, you have all patients who are in remission. So they are disease-free. And what you can see is in this beta diversity analysis that we have a significant separation in microbial profiles between the red and blue. So we can discriminate between patients in active disease or in inactive disease based on gut microbiome composition. Okay, and when we look at that was again reflected in the community richness and diversity. And so you can see that in patients who are in active disease, they have significantly lower microbial richness and diversity. And I hope you're familiar with these terms. Richness and diversity would describe the community composition, and Usually, a healthier microbial ecosystem would have higher richness and higher diversity, because when you have higher richness and higher diversity, you have more bacteria which can perform more functions and eventually can lead to a more healthy community. I mean, in a simple words, of course, there are many more aspects to these terms, but just in a very simple explanation here. And so we saw now that patients who are in disease have much lower richness and diversity, and they have also a very different microbial profile compared to patients who are in remission. And that was a very important finding, because that means that we're able to potentially to identify a microbiome signature or a pattern which we can use to describe a patient who is more susceptible to disease, or a patient who is probably will respond to a treatment, or will not respond to a treatment. So we can use it for a diagnostic and treatment purposes, for example. And so what we did next was that we used the information we have on the microbiome profiles, and we used machine learning tools to see if we can classify patients based on their gut microbiome composition. And here you can see in this receiver operating curve, or ROC analysis, so the higher the area under the curve is, the more accurate your prediction is. And here what we found was that we could classify patients into active or inactive disease based on gut microbiome composition with an accuracy of almost 80%. And based on this model, we identified 18 features, or 18 operational taxonomic units. Operational taxonomic units would describe bacterial species. So we identified 18 bacterial species which can be used to classify patients into patients in disease or patients in no disease. Okay, that's very helpful, but 18 bacteria is really a big number. If you want to develop a diagnostic tool, you would like to have the minimum number of features to use for classification. So what we did next was that we wanted to add additional layers of information. So not only the taxonomic composition of the gut microbiome community, but we also wanted to look at the functional potential and functional activity of these bacteria. So to do that, we did untargeted metabolomics. And in untargeted metabolomics, we basically look at the whole repertoire of metabolites in the ecosystem. So all metabolites which are secreted by bacteria. And here in this volcano plot, you can see that we identified here 332 and 119 metabolites which are enriched in patients with active or inactive disease. And we actually found out that most of these features are related to sulfur metabolism. And that was already one extra layer of information. So now we know that we knew that sulfur metabolism might be important or might be relevant to describe changes in gut microbiota composition or to inflammation in inflammatory bowel diseases patients. And so the next we looked here at these data are actually from predicted beta genomes. So using tools like PyCrust, we looked at the functional potential of the bacterial community in patients who have active or inactive disease. And we identified metabolic pathways which are differentially abundant in each case. And what you can see is that we also, to our surprise, we found some metabolic pathways which are involved in sulfur metabolism. And that was, again, a hint for us that maybe sulfur metabolism and sulfate-reducing bacteria would be relevant as a microbiome signature in disease. Okay. All right. So now, next, as I told you, now we identified almost microbiome signatures. So we knew that we have a difference between diseased and undiseased and healthy, and we have probably a relevance of sulfur metabolism and sulfate-reducing bacteria in inflamed patients. And what we wanted to do next is to do the functional validation using the humanized mice I introduced you earlier. So here what we used was IL-10 deficient mice, and this is a mouse model of chronic colitis. Under germ-free conditions, these mice are completely disease-free. However, when they have under specific pathogen-free conditions, or when they have complex microbial communities, they develop inflammation in the colon spontaneously. And so what we did here is that we selected 3 patients from our cohort. And these patients, as I told you, we followed over time, so up to 5 years. And we had multiple samples over time. And from each patient, we selected 2 different samples that we would take and use to inoculate germ-free mice and to test their functional impact in a mouse model. So, for example, from patient 16, we picked 2 samples, one in remission, one when the patient was in relapse. Here at baseline and when the patient was not responsive, or at baseline and when the patient went in remission. And what we did was that we took the gut microbiome samples or fecal samples from these patients, and we inoculated it in germ-free mice. So basically we do that by oral inoculation. So we prepare the fecal material in a certain way, and we inoculate it in the mice using a special syringe via the oral route. And these bacteria would go inside the mouse and would colonize the mouse. And we let these mice stay in these special isolators for 4 weeks. So they were colonized at 8 weeks of age, and we sampled them at 12 weeks of age. And the idea, or the reason why we did that, was that we wanted to see how a microbiome, which is originating from a patient at different disease state, would impact the physiology, or would impact the host in this mouse model, which is a mouse model for colitis. So it should mimic genetically the human IBD patient. And so what we did here, we colonized these mice, and to our surprise, what we found was that these mice actually reflected the disease phenotype of the donor patients, which means when we colonize the mice with gut microbiota from patients during active disease, the mice were inflamed. And when they received gut microbiota from patients during remission, they were disease-free. So that was fantastic, because that means that we can now look at these 2 mice, now these are, we had for each situation, we have 6 mice per group, and they were all, it was all reproducible. So we always had the same result when we inoculated a disease-associated microbiota. We had diseased mice, and when we had a disease-free microbiota, the mice were completely disease-free. And we looked at the mice in at different levels. So in histology, here you can see how an uninflamed mouse looks like, how the gut in an uninflamed mouse looks like. And here in inflammation, you can see thickness of the muscular layer. You can see a lot of immune cell infiltration, blunting of the crypts, and villi. And we saw inflammation in terms of also inflammatory cytokines and at the level of immune cell infiltration in the gut. So that was again a proof that the gut microbiota can transmit disease when transferred into a germ-free mouse. And what we did next here, we also did 16 sRNA gene sequencing on the mice and the respective human donors to see how similar the gut microbiota in the mouse is to the respective human donor. And what you can see here, so the arrows indicate the human donor, and in these circles you have all the germ-free mice which received gut microbiota from this one donor. And what you can see is that the mice which received one microbiota are always closer to their original donor. And this is the first conclusion. The second conclusion was that you can have gut microbiota which reflect different disease states. So, for example, in blue, you have mice which are colonized with gut microbiota from a patient in remission, and in red that was a patient in relapse, and in yellow that was a patient here that was a patient at baseline, and in yellow that was a patient in relapse. And so, as I told you, first conclusion is that always mice reflect the community composition of the human donor. So always the donor is closer to the mice, to the respective mice. The second conclusion was that you can have gut microbiota composition that reflect a remission state. So for example, blue, that would have a complete different microbiome configuration. So these two should represent a healthy microbiome, but actually they are also very different. So that's a beta diversity, and you can see that these two clusters are quite distant. So that means that you can, in terms of taxonomic diversity, you can have different communities that would confer the same function in the end. All right, and so what we did here in the mice, we again used machine learning algorithms to see if we can identify bacterial taxa that would be responsible for the separation between inflamed and non-inflamed mice. And here we identified a set of 10 taxa. And these taxa were able to classify the mice into inflamed and non-inflamed with an accuracy of 100%. So it was always right. Whenever you have a mouse which had an overabundance of disulfovibrio, for example, this mouse would be inflamed. And if you have a mouse which has a more, for example, Bilophila here, then this mouse was non-inflamed. So that was great because we said, okay, now we have a set of bacterial taxa that can identify with a 100% accuracy if the mouse is inflamed or not. And interestingly, one of these taxa was disulfovibrio. So again, a sulfate-reducing bacteria, again, confirming our findings, what we had identified at the level of shotgun metagenomics and metabolomics. And to confirm these findings, since now we identified that sulfur metabolism might be relevant, we did untargeted metabolomics in the mice, and we saw indeed that we have higher enrichment of sulfated metabolites in the inflamed mice compared to the non-inflamed mice. So the sulfated compounds are in the closed circles, and we had significantly higher richness or higher higher levels of these compounds in the inflamed mice compared to the non-inflamed mice. And then we confirmed these findings using targeted metabolomics, looking at certain bile acids, sulfated bile acids, and we identified some of these bile acids, which would again classify the mice into inflamed or non-inflamed. And finally, we used all these data from humans and from humanized mice, from 16S RNA gene sequencing, from shotgun metagenomics and metabolomics, and we integrated all these data in an integrative framework. And we used a machine learning algorithm to see if we can predict, based on this signature, the disease activity of the patients based on the signature. And if you remember what I showed you in the beginning, when we used only data from 16S RNA gene sequencing or the taxonomic diversity, we could identify or we could classify the patients based on disease activity with an accuracy of 80%. However, now when we combined or we integrated all these data sets from humans and humanized mice, we could identify, we could actually reach an accuracy of 96%. And we identified this set of taxa, including the sulfur Vibrio and E. coli, as the top taxa based on all these data we had integrated now in humans. And now basically what we do is that what we're currently doing in our lab is to look at the role of sulfur metabolism and IBD pathogenesis, and to look at the role of sulfate-reducing bacteria, such as the sulfur Vibrio in inflammatory bowel diseases. So with that, that's a summary scheme of what we did in this paper. Was that we started from a patient cohort that we recruited or was recruited with our partners in the clinic, and we followed over time. We collected fecal samples, mucosa biopsies, but here we focused on stool samples, and we looked into the gut microbiome composition and function using different tools, so 16S and shotgun metagenomics and metabolomics, and we confirmed these findings to look at the underlying mechanisms using germ-free mouse model. And in the end, we managed to identify this microbiome signature, which can be used to classify patients based on their gut microbiome composition and function. So just to give you a little bit of relevance, clinical relevance of this question. So imagine a patient would go to the physician and he would ask him to look at his gut microbiome, to analyze, to take a sample, stool sample, and to analyze it. And he would just look at the stool sample and or the composition of the gut microbiome, and he would instantly tell him, okay, you have a higher chance of responding to this treatment based on your gut microbiome composition. So that would be the application of having microbiome signatures for diagnostic purposes, for example. So in summary, what we found was that there is individual variations in the patients, and there are dramatic fluctuations during disease course. and that the humanized mice reflect dysbiotic and phenotypic features of the human donors, and that we identified sulfated metabolites and sulfur metabolism as a important or a key factor in the classification of patients based on disease activity, and that this microbiome signature could identify or could classify patient with an accuracy of 96%, and so it might have a relevance to the cause of disease eventually. And finally, just showing you this one summary scheme, which we have also in our recently published review in Nature Reviews. And what we propose here is that we need to move beyond correlations to causation. And to do that, we need to start from looking at patient cohorts, at large population studies, to do microbe and host profiling. And once we have these high throughput omic analyses, we can have biomarkers that we go back and do functional characterizations, for example, in mouse models, using or to do intervention studies. And eventually, we would have a validated microbiome signature that can be used for purposes for diagnosis and treatment of different diseases. And with that, I'd like to thank my team, the entire team at the Technical University Munich, and all our collaborators. And I'd like to thank you for your attention. And if you have any questions, I'll be happy to take them. Thank you.